Hello, everyone. Welcome to the GSA webinar, When Does Aging Begin? Part 2, organized by NIH in conjunction with the GSA 2020 Annual Scientific Meeting online. The webinar is being recorded and will be posted on the GSA website. A notice to all attendees will be distributed once the recording is available. A question and answer session will immediately follow the live presentations. But throughout the program, we invite you to send questions through the questions feature accessible on the GoToWebinar panel. So without further delay, I'm going to hand the microphone to Dr. Ron Kohansky. Ron? We can hold one moment. Looks like we've got just a slight technical glitch, not to worry. Can you hear us now? Let's see here.
He's logged on 3 times as a panelist.
Yeah, it does look like that. It might be, the system might be confused.
I think he should call back in once, log back in once, so he doesn't have 2 other
Yeah.
2 meetings that are muted.
Yep. Ron, if you like, we maybe we can start the program with Alyssa, and then when you come back on, we can just sort of shift the sections a bit. But yes, looks like you're in the system 3 times, but maybe if you log out and come back in, it'll clear that out.
Okay. I'm happy to go first, and then Ron can do a post introduction of the session.
That works.
Okay. All right, so can you see my screen?
Thumbs up.
Okay, welcome everyone. This is a really wonderful seminar that NIA, Ron Kohansky, and GSA have organized on when does aging begin? So we are 2 of 3, and I'm going to talk about early life, and Ann Newman is going to talk about during the lifespan, midlife and later life. And so we're really focusing on this question of when does aging begin, and when can we see it and measure it, and when does it predict outcomes we care about, early disease, mortality. And so I'm going to be talking about both trying to address this question as well as explaining the role of stress in aging. So everything that I love to discuss is really represented on this slide. So here, this is a bit of a summary of my view. So when does aging begin? It depends on when you look at the organism. So what stage of life are they in, and how can you see aging? It differs by stage. It differs by where you look. What level of analysis? Are you looking at molecular or cellular level indicators, or clinical risk factors that don't really show up till later in life? And what outcome do you care about? That will determine how much you care about the earliest measures of aging or not. So if you want to understand strong prediction of disease and mortality, then we're going to be focusing on later in life. We know that clinical risk factors, which are more proximal to disease and mortality, are always going to win as the strongest predictors. And the basic aging biology measures are going to be weaker because they are farther from the disease process. They're the components, predictive components of disease. I'm going to focus on telomere length. And that is because, well, we know a lot about it. It is one of the hallmarks of aging that's associated causally with, interdependently with many of the other hallmarks of aging, the basic cellular mechanisms of aging, but especially because telomere length is uniquely tracking throughout the lifespan. We can measure it on day one of life in cord blood, and we actually can predict offspring or a new baby's telomere length based on not just genetics, but on aspects of the social, psychological, and physiological environment of the mother and some aspects of the father. So uh, we and then my next point is probably the most interesting, which is the earliest biological aging begins probabilistically even before conception, based on who the parents are and the parents' gamete health. So of course genetics, beyond genetics is I'll be talking about the modifiable aspects of aging, and the sperm and the egg's health is transmitted. So it's not just the genes, but the epigenetic marks and the telomeres. They're directly acquired or transmitted in epigenetic-like fashion. So that is one unique aspect of aging that's transmitted and that tracks through life. And telomere setting and early in life is the biggest determinant of telomere length later in life. So it does change, it is modifiable, but what we start with as kind of initial setting point is quite important. Telomere length in midlife predicts robustly across meta-analyses early disease and now mortality. Small effect sizes, again, this is a distal molecular biomarker that we can measure at birth, but it is a consistent predictor. Aging also begins at ground zero. We had heard beautiful talks last seminar, both by Gladyshev and Belsky. And Gladyshev showed us a very elegant model of how early on in embryonic development, there is a kind of a resetting and a rejuvenation of kind of youngest biological state. And then at some point, the embryo starts aging. And that's when, he points out, that's when telomere length starts shortening. So that's another point during fetal development that biological aging begins. Some aspects of aging biology are most accelerated during childhood, during this influential sensitive period, and that applies to telomere length as well. This is childhood is the period in life when we have the largest rate of decline in telomere length. And then lastly, toxic stress accelerates biological aging during these critical and sensitive periods through the lifespan. So now I'll quickly go through slides to support these points. I love this model by Terry Moffat showing that we used to mostly focus on looking at elderly people when trying to understand aging. Now we understand that the roots of aging are in early life, and our more innovative models look at childhood risks, adversity, risk and resilience, pace of aging, which we can now measure based on multiple markers, as discussed by Dan Belsky. And I would have just added the fetal period into this graph here. So I also like this by Steve Horvath, which shows that as we age, we can look at many different levels of analysis, but the DNA level, the epigenetic marks and the telomere length are markers that early in life can be a metric for biological aging, whereas we don't see damage or aging in the organs and organism until later life. And then lastly, in this lovely model by Ferrucci, this makes the point, we can look at these different levels and they tend to show decline in age. Biological aging shows the earliest decline, but I would just argue that that first chunk of time early in life, we see the most decline. We see the most dramatic changes in biological aging, both during, according to telomere length and possibly epigenetic clock, during fetal development and early childhood. Okay, so in terms of stress, I have been studying stress for many years, and everything I think about acute versus chronic stress is here in this paper that just came out. which basically, talks about how when we think about acute stressors, they're inherently bad. They can even promote cell cleansing, housekeeping, and longevity. So that's the kind of the idea in the hallmarks of aging, that adaptation to stress, resilience to stress in the cell is a very important integrative marker of biological age. And in humans, unless we are losing our resilience processes, we can survive acute stressors. They can be salutary. And it's when we develop chronic stress, when we develop ongoing toxic stress for years that we don't recover from, that we see an acceleration of aging. And so this shows what this might look like over the lifespan, where ideal level of engagement in life and positive stress leads to an optimal biological aging and lifespan, whereas toxic stress leads to accelerated lifespan. So there have been many different studies showing that chronic stress exposure is associated with accelerated biomarkers of aging. That applies to telomere length, shortening, inflammation, epigenetic clock, and that's during adulthood. And what I'm going to suggest is that many of this is actually happening early in life. This is a study by Martin Picard showing that caregivers have poor mitochondrial function. And so yet another way that we can see all the way down to the cellular level, the stress acceleration of aging. So if we think about effects, we know in older adulthood, we lose some biological resiliency. So it's not that we're still vulnerable to the effects of stress, but in terms of the trajectories of biological aging, these are I pose most impacted in utero and in early childhood. In the public health literature, we now know that ACEs, early childhood adversities, particularly psychological trauma, is associated with early disease. But when we look at the biological markers of aging, we see that these are accelerated early in life. So there is biological embedding of toxic stress early in life that leads to telomere shortening, inflammation, and accelerated epigenetic clock. Now, back to focusing on telomeres as a barometer of age from day one of life, or from fetal development. This is a beautiful model by Sonia Entringer, Patik Wadhwa, and colleagues, showing the fetal programming effect, maternal states and stress during pregnancy, from psychological, behavioral, and health factors are shaping fetal programming, particularly oxidative stress and the HPA axis overactivity, impacting both placenta as well as the baby's hemopoietic system. So newborn infant telomere length is affected by maternal stress. This has been shown in many studies now. I want to point out a fascinating relationship. We don't know how large this is, and that is that beyond the imprinting of stress on telomeres during pregnancy, short telomeres are directly transmitted to the next generation. There are several genetic studies, like this one by Colapi and colleagues, that show that a parent with very short telomeres, due to a genetic disorder, passes on short telomeres to all of their offspring, not just the ones who inherit the genetic disorder, but the ones, the AA you see in the offspring with the Turk mutation to their telomerase, they have a premature aging phenotype without inheriting the genetic disorder for short telomeres. And that's because they directly inherited short telomeres on the gamete of the parent who had the disorder. What does that mean? It means that we may, through life experience that shortens our own telomeres throughout the body, including gamete telomeres, be transmitting stress-related shorter telomeres to the next generation. So you can see in a simplistic model, if there's an epigenetic direct transmission from parental germline to offspring germline. This has also been shown by Carol Greider. And I love this model by Hausman and colleagues that shows both the direct and indirect effects. Parental stress may affect parent germline telomeres, And then there's these indirect effects from the prenatal exposures and post-birth exposures. And parenting is important even from these ecologists. We know in the human literature that sensitive parenting is very important is an important correlate of telomere length. So I've told you that the earliest influences on telomere length come from direct epigenetic-like transmission of short telomere length from the parent, whether that's through life experience or poor health, or their own birth telomere length, prenatal programming, and early childhood stress, which casts a long shadow on telomere length. We can see the effect of childhood adversity on adults, healthy adults, even without depression. They tend to have shorter telomeres. So this lifespan model is that during pregnancy, prenatal stress can shorten telomere length, as can early adversity. Onset of depression, which happens in adolescence, is an important determinant. HPA axis is one of the mediators. And then in adults, what we see is correlates such as less neurogenesis in the hippocampus, lower hippocampal volume, and possibly these stress effects may affect the gametes as well. We know stem cells in their niches are semi-protected, but they're fed by the same blood source. So stress can affect them as well. Lastly, I've focused on stress, and I'd like to just span out to point out that the umbrella for stress, the body doesn't necessarily differentiate between psychological stressors and other toxic stressors. And so physiological stressors are becoming more common. Air pollution is associated with telomere length in adults. Maternal exposure to air pollution predicts shorter offspring telomere length. And this is something I feel that we can all be expanding our discussion to include how the climate changes that are here and that will be increasing are interacting with aging mechanisms. So life experience and chemical exposures are transmitted through generations, in part through telomeres and likely through epigenetic mechanisms. So aging starts both at conception, probabilistically before conception. Can we reverse these effects of stress? When we think about prenatal programming, we can. We know that prenatal programming or the early poor health exposures for women during pregnancy affects multiple calibration of systems of aging and predicts early disease. And so in this study, I'll just briefly tell you about, we have a intervention to work on metabolic health. So it's a nutritional, mindful eating intervention, and it also works on stress reduction. And this was co-led by Nikki Bush, who's been studying the offspring of these mothers. We had 200 overweight or obese, low-income mothers with high stress, and they went through an 8-week stress reduction intervention. We are still looking at their telomeres, so I can't report on that, but I can tell you that we saw many important effects that were co-benefits to both the mother and the offspring. So we found less infant illness, a healthier profile of autonomic reactivity of the infants, and less odds of rapid infant growth, which predicts lifelong obesity. We also, for the mothers, reduced maternal stress and depression, improved their glucose tolerance. So the bottom line here is this is just one example of a maternal health and mental health intervention during pregnancy. Pregnancy is a critical period to intervene and should be thought of as an anti-aging intervention for the population, for adults and the next generation. So in summary, aging begins in the womb. Telomere length is exemplary as a biomarker because it's directly transmitted from germline and shaped in utero. So it represents one of the earliest ways that aging is both transmitted and tracks throughout life and imprinted by our life experiences. Psychological and physiological stressors shape the rate of biological aging. I didn't go into it, but in the Aging Reviews paper I showed you on geroscience, I also discussed the positive benefits of stress on reducing biological age when it's short and hormetic. Pregnancy interventions should be part of our discussion. There are powerful anti-aging interventions that can improve health of multiple generations and could potentially be protective from the prenatal stressor effects of telomere shortening, yet to be determined. Thank you so much. I've ended with 20 extra seconds. Ron, do we have Ron?
Here I am. Can you hear me now?
Yes.
Yes, so thank you very much, Alyssa. It's always provocative talk and very informative when you give them. And I cannot actually see questions that are incoming, if there are any, at this point. So I'll just ask a couple on my own, if you don't mind.
Absolutely.
Okay, so there are failures of development that result from the same set of molecular disturbances, genome rearrangements, especially short telomeres, and stress. All of these can cause fetal development to fail. Do you see those as also as aging in utero?
So the type of aging I was talking about are, for example, epigenetic clock and telomere length are slowly, are programmed, calibrated aging systems that then unwind slowly through the lifespan, through the normal aging wear and tear mechanisms that interact with the environment and with life experience. Now, what you're talking about may be resulting in early death or developmental disabilities. I'm not, why don't you explain more?
Well, it's since they have the same, many of them are the same molecular origins.
Mm-hmm.
Asking if a distinction can be made between failures of development and failures that appear at older ages.
It's a great question, and there may be so different early mechanisms or causal reasons for ending up with those calibrations and risk patterns, but they may play out to look like early aging with the same mechanisms. And I'll just throw that back to you. What do you think, Ron?
I think that a distinction could be made between failures of fetal development because what's happening during fetal development is highly plastic. And one of the characteristics, I think, of older age is a loss of plasticity, which you can see in a range of functions. So I think that the plasticity of development is under selective pressure, but the loss of plasticity is more random and under substantially less selective pressure in evolutionary terms.
Yeah, I think that's a great answer. And I think that so we can differentiate between those pathways. But as Gladys pointed out, there is an early period of development, and after there's the blastule then we see the aging mechanisms kick in. So I think we can distinguish between those two periods, particularly in the more mechanistic studies, to really understand them differently.
Yes, absolutely. Thank you.
Thank you.
We'll have time for more questions later. Right now, I get the opportunity, since I can be heard, to introduce Dr. Ann Newman, who is Chair of Epidemiology at the University of Pittsburgh. Dr. Newman, please.
Thank you, Ron, and thank you, Judy. I'm pleased to have the opportunity to speak as part of this very interesting panel, and thank Ron for coming up with such a provocative question. While many do, when I just ask people casually when aging begins, have heard about the interest in early fetal development as part of the aging process, I'd like to talk more about midlife aging and hopefully to make a case that we really do want to distinguish between midlife and later aging and development. When Ron asked me to do this, I looked online for papers to hear and read about discussions that have been going on about this question over many, many years, and was interested to find this paper by Leonard Hayflick, who is well known for his publication showing that in cell culture, cells have a finite lifespan and develop arrested cell growth called senescence. This paper was a thought piece 20, 30 years after that discovery, where he did point out that we've known for some time that there are changes, there are accumulated damages that can be identified, not only at conception, but even at the gamete level in ova and in sperm stem cells that show that the process of biology results in an accumulation of damage because our repair mechanisms are imperfect. He also talked about, though, that many people do think about aging as the process after maturation, after sexual development, and the declines that occur, declines in function. And this is illustrated here, where we think about early life as being a period of development, adult life, and older age having declines, and that this is maybe where we want to focus on in terms of aging to maintain function after peak development. But as Hayflick said in this article, when aging begin, to answer that question, it really depends on how you define aging in the first place. So this is a definition that I use that's a composite of my reading over the years, that aging is the accumulation of damage and repair. It occurs at multiple levels, cells, tissues, organ, and in the person over time. And it results in a loss of function and a vulnerability to disease and death. And though we have experimental evidence that it's not inevitable, that it can be reversed, in general, we do consider it to be a process that's a universal condition of in biology. I think it's also very important to think about this damage and repair as remodeling, and highlight that by here some of the features that I want to talk about in terms of midlife aging, the damage and repair, the loss of function and vulnerability over time. So when we think about development, we think about cell differentiation and achievement of the maximal and very specific cellular function in different tissues is a feature of development that does not reverse in aging. It is maintained as much as can be after reproduction. Development is a period of growth, is also a period of remodeling, and in those remodeling processes, we see the same cellular biology in action as we see in aging. Development is also designed to maximize function. So I think it's appropriate to think about aging as the failed maintenance after that peak period of development. Another way to think about it, I've superimposed here the balance between damage and repair. And as I said, we all recognize that damage occurs as a feature of biologic processes in cells, including stem cells, including eggs and the sperm precursors. The balance between that damage and repair is such that we fail to maintain function after peak development. And you can think about that switch in that balance between damage and repair perhaps as the point at which after development we begin to detect a loss of function. I did have to amend this figure after reading, as Alyssa Eppel pointed out, Vadim Gladishev's recent article pointing out that it does appear that there is a rejuvenation period after fertilization, after fertilization, where the early cellular development in the blastocyst actually is a improvement and a cleaning up of damage that is present. And without this cleanup of damage, you would have accumulated that damage from generation to generation. So the idea that there's a biological process to reset to a ground zero seems to be timed to the transition from the blastocyst stage to the development of organogenesis. And I think that's a fascinating idea. It makes a lot of sense, and it explains why we don't inherit too much damage from generation to generation, though some of it is passed on. This is an illustration from that paper that shows in this period that the telomere length actually increases from fertilization to organogenesis and then begins to decline. And that rejuvenation period is a process that we might even think about intervening on to improve and optimize that ground zero period during fetal development. So again, as Dr. Epple has so clearly pointed out, this period is critically important in development for having a better aging process. But I think the decline in function after maturation is a more logical set point for defining the onset of aging. It's very logical. I think it has faced validity in describing the failure to maintain, but it's also something that we can begin to detect and measure in the whole person at a point where we probably would be most likely to think about interventions. It seems very unlikely to me that we would, at this time anyway, really think about treating the aging process from conception or birth, but more likely to modify the trajectory later in life. And this idea that I'm proposing, that we want to think about aging beginning after maturation, does take into account and a full appreciation for the fact that there are many overlapping biological processes that I think Ron's question clearly pointed out. So I'm talking about this period after peak development where there's decline, but it's slight and a bit of a plateau before you begin to see changes. So if you're going to look at aging there are a lot of important concepts in evaluating that period of time. First of all, it's really important to be able to detect the peak of development. And we don't really know if all systems have the same peak, but they don't quite seem to. For those that have been clearly measured, there's definitely a tremendous acceleration in development during puberty that's achieved by the late teens. But thereafter, there's a deceleration of that development, but it doesn't stop. And for many things that we look at, that development can continue very slowly after the late teens into even the 30s. So it raises the question if development can continue at the same time that what I'm saying, decline or aging, begins, and I think the answer is yes. And that's why it's so difficult to actually pinpoint when aging begins, because you have an overlap where development may be continuing, but for certain aspects of health, decline is beginning. A second issue in defining it is that the time course of it is very, very slow. We say it's about 1% per year, but it's actually slower than that early on, and it's faster than that later on. But it's still very, very slow and has a tremendous amount of variability. So there's a signal versus noise problem with detecting this. There's also an issue about intrinsic damage versus extrinsic factors. Some have proposed that aging should be the fundamental biologic processes that occur independently of injuries or obvious external damage. On the other hand, you can define those extrinsic factors as accelerants of the aging process, but I think it points to the value of studying healthy aging cohorts to really understand what's fundamentally going on biologically independent of external toxic drivers of aging. There's also issues in measuring with ceiling effects. Many of the things that we measure in people are designed to measure diseases, and they tend to pick up the lower end of the distribution and not be challenging enough to detect early change. And dementia tests are a great example, which have been used in midlife to try to detect early cognitive decline, but they're not challenging enough. Another issue is range effects, that many times the best value is a sweet spot. It's not either good or bad, it's somewhere in the middle that we can define. I'll show you examples of some of these. And finally, I'd like to discuss some of the adaptation that occurs and the challenge of measuring aging when there are so many counter-regulatory changes and adaptations. So in terms of detecting peak performance, one way that we do this is to look at the records. This is an example of records for marathon times. And like the sample figure that I drew, you can see here that time gets better and better and better until the late 20s, and it seems to plateau a bit. And so if you want to look for decline in something like this, you don't start to pick it up probably much until after age 40, at least on a population level. So where is the peak? It's going to vary for individuals. Here's another example of an attempt to find a peak by age in muscle strength. This study looked at grip strength across 12 different British studies and pieced them together. And here you can see a similar pattern of a peak continuing to be present after the 20s, and very difficult to pick up much decline until after age 40. Easier to pick it up in men than in women who have lower strength at all ages. A reason that this is so challenging is that there are huge birth cohort effects. This is a paper that looked at birth cohorts and their trajectories of multimorbidity. You can see that when stratified by obesity in the dark lines versus non-obese in the dotted lines, that the onset of obesity has been a huge accelerant in what you might call aging in the population in each generation. Income also has a major factor. So do we consider these income, aging, and other social factors as accelerants? If we do, there are huge birth cohort effects, so that if you look at any population at a single period of time, you're going to mix these factors. Another issue is that the decline that we see after age 40 is very much dependent on the peak. This shows the loss of VO2, which is exercise endurance, in groups that are at various levels of lifelong physical activity, where the top dotted line is the group who has maintained high physical activity throughout their life. And what's striking about this is that regardless of the physical activity level, the rate of decline appears very similar after age 40. And the track that a person is on is very much just dependent on the peak that they achieved. So this idea of peak development, determining the trajectory that one is on, is very important. And I think that it's different, because I think it's different biologically, it then becomes a different target. We looked at this digit symbol test as a measure of cognitive function in a cohort study of healthy agers. These are participants in the Long Life Family Study in the orange line. This is actually cross-sectional data. But what we see is that compared to a general population cohort study, their cognitive function is higher at every age where we had overlap in the age group. But when we have a broader age range in the Long Life Family Study, it looks like they're just tracking according to a higher peak to start with. So are they aging differently? It doesn't look like they really are. This is all due to them achieving peak development. Again, another reason to think about development differently from the failure of maintenance. Another issue is the variability in different organ systems. We don't think that these peaks and these declines exactly track for each system. And this is a figure that shows, that each color would be a different organ system. When we looked at this again in a healthy aging cohort, we found that the correlation cross-sectionally between different systems was very, very low, actually, that the different measures of kidney function, vascular function, lung function, metabolic function, muscle strength, that they did not seem to be on the same trajectory. Another reason that it's challenging to measure just one thing and understand aging. We also looked at healthy aging across phenotypes and families, and we hypothesized that the healthiest people would have the be healthy in multiple domains, so that what we found here, you see very low correlations between these phenotypes. We found that the families who were the smartest were not the families that were the strongest, and they were not the families with the best blood pressure, and they were not the families, by and large, with the best metabolic function. So there's this aspect of heterogeneity by organ system that makes it very difficult to define aging globally. Another issue I mentioned is that there's often a sweet spot for any given measure. Here's an example of how we try to define a healthy blood pressure in families across generations, that people whose blood pressure was too low or too high for their age were considered to have an unhealthy phenotype. And we found that most of the familial relationship of this healthy blood pressure was probably not driven by genetic factors. But the point is that if you just look at blood pressure as a single measure and you don't consider the fact that it varies in whether it's good or bad at any given age, you won't define a healthy aging properly. Another issue to think about is the issue of remodeling. Many of the things that we measure, we think about measuring things declining all the time, but in fact, as people age, they're having a tremendous amount of remodeling. This figure shows how different features such as higher blood pressure or faster blood flow or injury to the artery can greatly influence not just the thickness of the artery, but the width of the artery and the size of the artery. And there's increasing interest now in understanding not just measuring things going down, but measuring how they're remodeling. And in this example, there's a picture of a carotid ultrasound. This comes from work showing that the dilation of the artery gets bigger and wider, and that's a separate feature of aging that is independent of the thickening of the artery. So it's much more complex to measure some of these things and not just assume any one dimension of these complex phenotypes. This has been very well studied in the bone field, where as bone density goes down, the width of the bone also gets larger, like we see with the arteries. And these are independent features that influence risk. So if we're measuring aging in midlife, there are some things, though, that are pretty good at measuring the whole system. Aerobic fitness is something that does go down as people get older, as I showed you, even if they're very physically active. Muscle strength, more challenging cognitive tests, measures of vascular stiffness, which captures that arterial thickening as well as dilation, transparency of the lens of the eye, and just general stress responsiveness. And now there's tremendous interest in using blood biomarkers, epigenetic markers, telomere length, as Dr. Eppel has discussed, hormones, inflammation, metabolic factors and measures of early disease. And these have been put together into indices of biologic aging. Even these are challenging. Here's a paper from Dan Belsky that he did this wonderful comparison of telomere length, methylation clocks, and biologic age from biomarkers in the blood. It's very interesting that he found that they did not correlate well with each other. So again, this tremendous heterogeneity. I think that the reason for this is that the methylation clocks are tuned across the lifespan. They include both development, programming, and aging, and that's why they relate to each other, but they do not relate to the biological age, which use markers of damage and reflect this period of decline after development. So again, this is to me a reason why we should think about development separately, that I think if methylation clocks were based only on the post peak period, that they would be more closely aligned with these other measures of biologic age. So in terms of defining aging, many people have basically made damage as an equivalent to aging, that damage, cellular damage, that comes from biologic processes that are not perfect. This is aging. But I think that we should think about aging as vulnerability. Vulnerability is damage and repair, and we should disentangle the developmental peak from the decline, as well as consider tissue specificity. Now you're beginning to see methylation age for the immune system, methylation age for given organs, and they are different. And in terms of intervening, I think it emphasizes the need to prevent damage and promote repair to optimal remodeling. So I'd like to recognize the postdoctoral fellows, Jason Sanders and Megan Marin, who worked on the papers in the Long Life Family Study and the Cardiovascular Health Study, the other studies that I mentioned here, and the funding from the NIA and NHLBI. Thank you.
Thank you very much, Dr. Newman. Great presentation. I will take host's privilege to ask a question, of which I have many, but I appreciate the points about the difficulty in describing or defining what aging is, but it may be slightly more difficult to define healthy aging, which you and others have referred to. Would you comment a little bit more on what you consider to be healthy aging?
So yes, I agree that it is also very difficult to define. I think it's most easily defined after the fact when you see that someone has lived a very long time with minimal health challenges, without cancer, heart disease, stroke, etc. So how do you define it in sort of prospectively? It's easier to define it retrospectively in the longest lived people. So I think that anything that we develop for biomarkers of aging, they ultimately have to be validated against prediction of mortality and prediction of function. And I don't think there's a clear demarcation of what is healthy aging, but I would say that a biomarker of aging should be able to predict the best outcomes, the lowest mortality in a group, and the highest function in a group.
So that might mean then, considering in a population, if people are outliers relative age, let's say, or what is often called biological age, and if those outliers are not artifacts of some data collection error, they might be topics that would reduce the heterogeneity a little bit.
Yes, when you look at people in midlife, and I think that I wish Dan Belsky could be part of the discussion today, but when you're looking at people in their 20s and 30s for aging or for healthy aging, most people in that age range are going to ceiling out on most of the measures that we make in a whole person. So that's why there's so much interest in looking for circulating factors and cellular change that can uncover the very early origins of age-related decline.
Okay. So I'll go to questions from the audience. How does physiological decline correlate with our historical odds of reproductive success? Either of you care
Well,
to
I
take
think it's very clear that reproduction is most successful in people at the peak of their physiologic development. And when you have older parents, both fathers and mothers, you increase the risk of there being DNA rearrangement or deletion or something that will increase early fetal loss, but also result in a higher risk of poor birth outcomes. That's why older maternal age is considered a risk factor for poor birth outcomes, but it's also older father's age that's also a risk for transmitting more damaged gametes, I guess.
Yeah, so that harkens back to Dr. Appel's point about what we inherit will affect our aging.
Yes, so it's a great question. I'll just comment that when we can measure and look at ovarian aging, it's very important. It's very interesting as an early precursor to the rest of biological aging and the midlife aging measures that Anne was talking about. It predicts, for example, arterial stiffness and earlier onset of cardiovascular disease. Ovarian aging is obviously important for fertility, but it may be an an early organismic measure of health. We know that shorter telomeres in the granulosa are predictive of less fertility, worse birth outcomes. So how do we inherit short telomeres in our eggs? It all goes back to both the genetic and the acquired direct inheritance of telomere length. So I think it's a piece of it. And, it is complicated. There are sex differences. It turns out the older fathers are transmitting sperm with longer, not shorter telomeres.
Fascinating. I did not read for simplicity the second part of the question, but it did state for males, this may not simply decline through adulthood, but actually be higher at 35-year-old relative to a 20-year-old. Not that I have good data, but I will comment to say that on the gender difference or sex difference, Aristotle commented that the optimum age for reproduction, as they put it, marriage at the time, a male in his 30s and a female before 20. So clearly sex differences were appreciated 2 millennia ago, but not necessarily the biological underpinnings being known. There's other questions here. Given that older adults tend to report less stress, does aging tend to slow down with chronological age?
Go ahead, Anne. I'll answer after you.
Yeah, I think that there is evidence that there is a deceleration very, very late in life. It may be a survival effect, though, that we're looking at, a bias in what we're looking at.
It brings up the point to me, the lovely work of Laura Carstensen and others, that we do see this, robustness in mental health overall. And as we age, increases in wisdom, the decreases in the negative affect to positive affect balance, so shaping lives toward positive social connections, experiencing more positive affect, and not choosing to be selecting out of very stressful situations and social relationships that are stressful. So there's definitely a kind of a positive profile of psychological well-being during aging that is important. We also know that purpose in life as a predictor of both biomarkers of aging, like inflammation, but also more healthy longevity and health span. About the stress piece, my point was the opposite. So my point was that we have the critical period of development and the sensitive period of childhood when stressors have a deeper, more damaging impact on aging trajectories, on molecular-based measures of aging biology, and that during older age, we don't see dramatic changes in epigenetic clock and in telomere length. So those molecular measures are a bit more stable in the adult years.
Might that be because there's simply resilience has not yet developed adequately?
Yes,
Great.
so I think part of the definition of the toxic stress really is about level of resilience. So if we're responding to acute stress with both the physiological counter-regulatory processes that Anne mentioned, that's going to be a hormetic stress response. And so similarly, if our psychological coping resources are well developed, we can respond. The stressor will be more likely to be positive or hormetic than toxic. So it's about both physiological and psychological resilience reserves.
Okay. All right, I'll read one last question. I believe this is directed to Dr. Eppel. You discussed effects of the stress reduction intervention on the offspring. Did you see any effects on biological age of the mothers with these interventions to reduce stress?
Great question. We were looking at 200 overweight and obese women in that study. Pregnancy did not affect their telomere length, as published in other studies. The intervention did not appear to affect the women's telomere length. The amount of sugared soda, we did a dietary analysis, and sugary beverages led to decline during pregnancy, regardless of the intervention effect. The intervention did improve other important aspects, a better glucose response to the OGTT. It also led to reduced depression for a whole year afterward. And in terms of long-term trajectories of aging, mental health precedes physical health. So depression and anxiety, years of these psychiatric disorders, these happen earlier in life and they precede accelerated aging later in life. So we do think there's still, an effect of reducing the postpartum and midlife depression is going to be an important piece of slowing aging and anti-aging.
Great. There were two more questions in the that were presented, and I think those will be forwarded to our two speakers today, but due to time constraints, we need to wrap up and advertise the next session, which will be December 16th, 12 to 1 p.m. Eastern Standard Time with Drs. James DeGregory and Mark Miller. Once again, thank you very much, Drs. Eppel and Newman, for very stimulating presentations. And let me close by asking if either of you would like to make a few final remarks.
I appreciated the discussion and the question. I love, you're such so thoughtful about aging, Ron. Thank you for this. I'll just say it's not either or. I think that initial setting, fetal development are clearly determinants of aging, but Anne's points about aging after the peak of development are also so important. Again, it's about how strong of a predictor do you want of later disease? We don't know yet how strong these early childhood clock and telomere length predictors are of later life disease and mortality. No one has measured these in cord blood and followed samples out. So we need those. I loved Anne's point about intrinsic versus extrinsic aging. I think we definitely need more discussion of that. There's a paper by Terry Moffat on geroscience in aging research reviews that really explains how theoretically it's important, but biologically people are so complex that aging is an open system, and the extrinsic factors that affect aging have to be taken into account when we think about biology of aging.
And if there's time for me to add on to that, I would just like to say that the challenge that we have in thinking about rejuvenation or reversing or slowing aging is that when you think about development is differentiation, and what we don't want to do is de-differentiate our highly specialized cells, and this is why I like to think about them as two different processes.
Excellent. Thank you very much, both, and hope to be in touch with you in the not too distant future.
Thank you so much.
That concludes our webinar for today. Take care and happy Thanksgiving.
Yes.
Thank you.
Thank you.